my name is Emily, and today I'm going to talk about viral flow, which is a tool that can be used to infer viral spillover risk using metagenomic and wastewater data. So this tool is really aimed at improving pandemic preparedness, as we saw in the most recent pandemic, COVID-19, that exposed the importance of surveilling and being prepared for pandemics because the public health, social, and healthcare system effects are still apparent in the world that we live in. So we learned that being prepared for these pandemics and having surveillance measures in place is really crucial to avoid such detrimental effects to our healthcare system. And a way to surveil our environment for viral pandemic threats is to use genomics. So following this genomic surveillance strategy scheme here, we can take samples from humans, animals, our environment, and analyze them for viral signals that can be linked to clinical and metadata for analysis, and then look at reporting it to inform public health responses. And so this flowchart is a continual and iterative process for proper surveillance, and we can develop such genomic tools by identifying sources of potential viral threats to sample from our environment. And these viral threats may come from other animals and the environments that these animals interact with, such as bats that may consume wastewater. And this can lead to something called a spillover event, where viral genetic information is transmitted from a reservoir, such as wastewater, to a new host, like a bat or vice versa. And such spillover events can be a threat to human health, as we know that viruses can be transmitted from animals to humans through bites, consumption, etc. And so to analyze the risk of a spillover event, particularly between wastewater plants to bats, we can use metagenomics, where we take sequencing DNA reads from things like bat feces samples and analyze them for viral signals from known virus genomes. We can compare this to viral signals found in wastewater samples and assess the risk of high overlap between specific viruses in bats and wastewater samples. And so the goal of our tool Viral Flow is to integrate animal metagenomics data with wastewater viral concentrations to create a tool that can surveil geographical spillover risk. And we imagine this is mostly going to be used with public health officials to assess, again, priority locations that may be at risk for viral spillover events. And so the pipeline of Viral Flow starts with required input, which is a FASTQ read file that contains our DNA sequencing from animal samples, again, like bat feces, as well as the sample coordinates from where that sample was taken. And then we want to identify viral matches from the reads compared to known reference viral genomes. And with Viral Flow, this can be done at two different levels. You can use config mode, which basically tries to piece together these reads into a long contiguous DNA and reconstruct the original sequence that then can be used for higher accuracy matching to the viral genome. Or you can do sort of a quick, dirty, noisy way using k-mer selection, where we divide these reads up into smaller sequences of k length and try to match these against the viral genomes. But once we have a viral hit, we've identified a virus that's supported by our reads data, we can then compare this virus to viral concentrations measured in wastewater data. And this is then used to calculate a spatial risk score, which is a composite score that flags locations, these wastewater plant locations, for further surveillance of a spillover risk. And so sort of under the hood of Viral Flow as well, there's a database that supports all of these data usage cases. But more importantly, what I want to talk about is the interpretation of the spatial risk score and how it's computed, because this really is going to flag locations that should be further surveilled for spillover events and where we imagine public health officials interacting with Viral Flow the most. And so again, this is a composite score, which is the product of 4 different components. So the score for each plant P and virus T incorporates a animal viral signal strength, which looks at the relative abundance of the viral signal from the reads, as well as the number of match reads and the confidence intervals of the abundance. And then similarly, we have a wastewater viral strength signal that looks at the current viral concentration from the most recent collection of the wastewater sample, and then also the fold change against a historical baseline from previous data. Then we also take into account some more epidemiologically focused components, like the population weight, which looks at the population served by a wastewater plant. So this is going to boost risk scores for wastewater plants that serve more people, as we expect a greater impact from a potential spillover effect in wastewater samples. And then also a distance factor, which downweights samples that were collected farther away from a given wastewater plant because we expect less direct contact between those two different sources of viral signal. And so this score is then scaled between 0 to 100 and can be categorized for high, moderate, low, or minimal risk for further surveillance, which then can, prompt further public health response. And so we really find the use in this that it's a single value that sums up these multiple components that can be easily interpreted for public health officials to inform, again, downstream public health decisions. And so the main output of viral flow, again, that we expect public health officials to use is this interpretable output file, which pretty much in the name is the most interpretable output file that viral flow produces. But there's a lot of information here that can be used if that's of interest to a user. Like here we list the different viruses that are overlapped along with their corresponding statistics. But the most important part of this file is this overlap table, where we list the different wastewater plants along with their risk score categorization, their score, and the virus that overlapped. And so no surprise here, we used bat data, so we have a lot of SARS-CoV-2 with very high risk. But overall, this table can be used to flag these wastewater plant locations between the animal and wastewater viral overlap, and again may prompt further public health investigation and surveillance at these locations. And so in testing viral flow, we demonstrate identification of SARS-CoV-2, again like in the previous file, using bat data among other viruses down here on the x-axis as overlapping in the bat metagenomic sample here in red. So these are the reads that overlapped with the viral genome, and the wastewater viral signal in yellow. So this is the concentration found in wastewater samples. And so this data was produced with a subsampled set of a total reads file. So we'd expect these numbers here on the y-axis to be a lot higher when processing a whole FASTQ reads file. But the utility here is that viral flow is able to extract these viral signal overlaps, again, between our animal metagenomic sample and the wastewater viral concentrations. And we also observed the effect of the multiple risk score components because viruses like SARS-CoV-2 and let's say HMPV have a similar overlapping, kind of, but their risk scores output by viral flow are very different. And again, this comes down to the fact that we are incorporating multiple different components in this composite risk score, things like distance, population, that put it in this greater epidemiological context beyond looking at just viral signal alone. And that's reflected in these difference of scores here. And so we can also use viral flow as a way to surveil regions to identify states with higher risk for further surveillance. And so here we used various different coordinates of sample origin from Colorado, California, Florida, Maine, and we use the same animal sample. So we want to test if different viral signals can be recovered using different sample locations. And so on the y-axis here, we're measuring the total number of viral animal wastewater overlaps, again, using subset data, so we'd expect this number to be a lot higher with a full file. But out of these states, we see that viral flow is able to extract different proportions of risk levels among these different origin sites, with California having the most high-risk overlaps, which again may lead a public health official to take a further action to flag that state for closer public health surveillance. And so in conclusion, we present viral flow, which is a tool that can be used to identify locations that may be at risk for elevated risk for a spillover event using this interpretable spatial risk score that's designed for public health official interface. And there are some important limitations to note about viral flow, one being that the database currently is static, so users have to update the wastewater plant data for future years and also provide sample coordinates, which are things that could be automized that we're currently working on. And additionally, the data that we showed today is only tested on bat metagenomic data, so this could be expanded into other species, things like livestock that we know interact with wastewater, to see what those results look like. Thank you.